A major field of interest in the study of aging is telomers, and telomers are like sticky ends on the ends of DNA. And if we think of a typical chromosome, we can think of the DNA as like this really long rope-like structure, and every time your cell divides, these sticky ends of telomers get shorter and shorter. And the consequence of that is, as your cell continues to divide, as you age, the less sticky the ends of the DNA are, the more frayed the rope, the chromosome, will become. And this leads to problems such as cancer, higher rates of cancer and other diseases as people age. And so in cells, we have proteins called telomerases, or telomerases, and their function is to rebuild telomers. And in healthy cells, these telomerases do not work. They're turned off because part of natural selection and evolution is you go through life and death. But in cancer cells, such as HeLa cells or HePa-1,6 cells, cancerous liver cells, this telomerase enzyme is reactivated and it's permanently turned back on, and that's what allows cancer cells to be able to divide and grow so voraciously in people. And so studying this telomerase enzyme is a big topic that a lot of people are interested in. And it's very easy in a lot of these bio classes to just do a lot of hand waving and say, if we can determine how to disable telomerase in cancer cells, we'll have fixed cancer. But in practice, things are a lot more nuanced and complicated than that. And so what we really have when we say telomerase is actually a complex of individual groups of proteins. And when people were studying telomerase, they knew one particular part, which was TRF1. And TRF1 was just one piece of the puzzle that is telomerase. To activate telomerase, to understand fundamentally what telomerase is, there are other proteins present, other protein complexes that we just didn't know existed. And what the yeast 2 hybrid system allowed scientists to do was discover a major component referred to as TIN2, TIN2, which was another piece that makes up the puzzle of telomerase, in addition to TRF2. And so when this complex of proteins comes together and assembles, we have telomerase, and we can much better understand the mechanisms by which cancer works. And the way we did that was with the yeast 2 hybrid system. And so understanding the yeast 2 hybrid system is very important in studying biology and cancer in particular. And so the way it works is we would have some kind of strand of DNA. And in this DNA, we would have some kind of region with LAC-Z or some other kind of gene that would allow us to perform a bioassay, such as green fluorescence protein GFP. And on this segment of DNA, and these are referred to as reporter genes, on this segment of DNA upstream, we would have an activator. And this activator is called it's called the upstream activator sequence. And I'll call it UAS for short. And the upstream activator is only activated when there is an binding domain and an activating domain, which are these protein complexes that interact directly with DNA, present. And so what happens in the yeast 2 hybrid system is we'll have the binding domain, abbreviated BD, connected to something referred to as the bait. And the bait is a known protein typically. So in the case of studying telomerase, scientists already knew about TRF1, but they had no idea about TIN2. So in this case, the known protein would be TRF1, and the binding domain would be specific to whatever your reporter gene is. In the case of LAC-Z, the binding domain was GAL4. And don't worry about remembering all these terms. It's mostly just understanding concepts for biochemistry classes. And if you're doing research, just having a good understanding of stuff is good to have. And so, so we have this one protein present in your solution along with your DNA sequence with your reporter genes. We also have present the activating domain, abbreviated AD, connected to your prey. And what will happen is the bait and the prey may or may not interact with each other favorably. And so if there's an affinity between the bait and the prey, the binding domain and the activating domain will be will activate the UAS. So to draw this out on your promoter region, your UAS, we would have the binding domain sticking onto the bait, which would be some protein we can draw a little bit like Pac-Man. It's a very poor Pac-Man. But if your bait had a match for the prey and an activating domain, if these two matched up correctly, the binding domain and the activating domain would be good enough to allow something like RNA polymerase to come by and transcribe the reporter gene, which would allow us to have some kind of physical evidence that there was this interaction between the bait and our prey. And to analyze the genomes that are present, in the, so if we're looking at the cloning vectors or the plasmids, we would have the binding domain here and the bait gene here, and we would know the bait gene a priori, but with the activating domain and the prey gene, this prey gene is something that we would get from genetic diversity, which is a fancy way of saying error-prone PCR, or some other mechanism that generates DNA fragments with lots of mutations. We would insert that into a cloning vector and then translate that random sequence of DNA into a protein, and then see if that DNA had, or if that protein had any favorable interactions with your bait. And if it does, then we will have discovered a new protein complex that is very important in the field of oncology. And so just to recap, the bait in this case was the TRF1 protein. The prey, after several iterations, would be the TIN2 protein complex that we just discovered. And from that, scientists were able to discover a major component in how telomerase works. So it's pretty cool stuff if you're interested in how cancer treatments are coming along. And for me, I think the really cool part is just how the scale that we are able to run these experiments on, because we're essentially throwing stuff at a fridge and seeing what sticks and just doing it again and again and again. These kinds of air-prone PCR things that generate genetic diversity, and then we see what sticks to our bait. We're essentially doing something equivalent to just brute force guessing every protein that exists in the book until we find something that has some kind of affinity for a known protein. And then we, once we figure that out, we can move another step down the chain and continue this process of discovering how these protein complexes are actually, what they're actually made up of. So I think it's pretty cool stuff. Let me know if you have any questions, and thanks for watching.